I'm Carolyn Hiller, the Program Director for the Clinical Diagnostics Program. And for those of you not familiar with MDIC, the Medical Device Innovation Consortium was the first ever medical device public-private partnership between nonprofits, industry, government, and that includes the FDA, CDC, CMS, and NIH. I'd like to introduce our panelists today. We have Annie Saha, who's the Director of External Expertise and Partnerships at the Office of the Center Director. We have Marina Kondratovic, who's the Associate Director for Clinical Studies in OIR at CDRH. We have Giovanna Tezek, who's Associate Director for Science and Technology in OIR at CDRH. And we have April Viokas, Director of Regulatory Affairs at Abbott, and she's also our working group, our Surrogate Sample Working Group Chair. We'll reserve the last 20 minutes for Q&A. You can enter your questions in the chat box down below at the corner of your screen. And now I'd like to turn it over to Annie, who will give you additional information on the public-private partnership.
Good afternoon, everyone, and I'll keep mine brief to get us sort of back on time. As Carolyn noted, MDIC is sort of really the first public-private partnership to focus on regulatory science in the medical device industry, and really what can we do to develop tools and methods and other sort of pre-competitive information that's going to help the entire medical device ecosystem innovate and ultimately lead to better diagnostics and therapies for patients. And this public-private partnerships, really a nexus of government, industry, and nonprofits, including patients, providers, and academics. And really it's that whole concept of that the sum is greater than any of the one parts, and if we all come together and work together, we're really going to be able to move things forward much more quickly than if we each were to do so individually. And ultimately, it's an alignment of both resources, people, and intellectual capital. Next slide. In terms of some of the other initiatives that MDIC is doing, just to give you folks a background, and the focus, of course, of today's webinar is on the clinical diagnostics work, but there's certainly other work streams that MDIC is involved in that may be of interest to you. In terms of clinical trial sciences and computational modeling, there's been a lot of work in terms of developing things like virtual patients and how you can use virtual patients in your clinical trials as Bayesian priors instead of necessarily, and potentially in lieu of enrolling actual patients. There's been a lot of work in the Case for Quality initiative looking at maturity models and metrics and how do you look at quality, and MDIC has been an integral part in a lot of FDA's effort in looking at what's critical to quality and the pilots that we are currently doing in the Case for Quality space. In terms of science of patient input, MDIC has done a lot of effort and really sculpted the fog, if you will, in terms of patient-centered benefit risk and developed a framework for how to incorporate patient preferences in regulatory decision-making, as well as a catalog of methods of how to use preferences all across the entire device product lifecycle. MDIC has also been awarded the NEST Coordinating Center, NEST being the National Evaluation System for Health with a small h technology. And the idea behind NEST is really how do we develop a better clinical evidence generation system and really pull together the pre and post-market data sources and weave all that information together in a way that we have robust both pre-market data generation as well as post-market surveillance and really sort of break down the silos between data. And ultimately, all of these projects, and of course the clinical diagnostics work, is ultimately what we're all trying to do in the ecosystem to benefit patients. And I'll turn it back over to Carolyn now.
Carolyn, if you're speaking, we cannot hear you.
Enter your access code or meeting number followed by pound. Enter your attendee ID number followed by pound. If you do not know your
Hello, can you hear me now?
Yes.
Okay, yes. My apologies for that. I'm not quite sure. Well, it seems when I put my phone on mute, it stays on mute. All right, so our clinical diagnostic projects that we have underway include the surrogate sample. You're going to hear about the framework, and we're currently working on harmonized educational materials. We have a clinical evidence white paper that is looking at the evidence that needs to be developed for analytical and clinical validity as well as clinical utility. And we're also working on an IVD real-world evidence framework that will look at applying real-world evidence as a source of evidence for regulatory support and reimbursement decisions. And we also have our fingerstick blueprint project that focuses on developing study design blueprints for using fingerstick capillary blood draw for analytical validation of tests. Now I'd like to turn it over to Marina Kondratovic from OIR to briefly share how the surrogate sample framework will help the FDA.
Okay.
Marina?
Thank you very much. Ideally, one needs to use patient samples from the intended use target population in all analytical studies, because mainly patient samples provide information about performance of the assay with the patient samples. But frequently, the needed patient samples are difficult to find. For example, in comparison study for estimation of biases, we need to get patient samples which should cover entire measuring interval. And it is not easy to find patient samples with high values of the analyte. Or sometimes it's even impossible to use patient samples in the analytical study. For example, precision study requires measuring the same patient sample under different testing conditions. Very frequently, it is impossible to do this using a patient sample. Surrogate samples are used in all FDA submissions, and in every submission, there is at least one analytical study that needs to use surrogate samples. So a use of surrogate samples is not really a new topic. But there are not a lot of information about scientific questions related to this issue. Next slide. We all know a basic principle in using surrogate samples, that surrogate samples should mimic patient samples as much as possible. But details about how to use this principle were undeveloped, and we hope that the framework about surrogate samples provides these details and presents a step in development of laboratory medicine science. There are a lot of questions, as for example, for different analytical studies, different characteristics of the tested samples are important. So surrogate samples, which reflect the patient samples in the best way for one analytical study, may be not the best surrogate samples for another analytical study. Which characteristics of the surrogate samples are important for this particular analytical study? This framework helps to answer this question. Another example, which we just discussed, in the comparison study for estimation of biases for the quantitative assay, we need to have patient samples which should cover entire measuring interval. And it is not easy to find patient samples with high values of the analyte. Is it better to prepare surrogate sample as, for example, first approach, to pool all negative patient samples and then to spike this pool with few high values concentration? Or maybe second approach is better. Maybe it's better to spike individual negative patient samples. So this framework helps to answer this question also. Next slide. We think that the surrogate sample framework is beneficial for manufacturers, reviewers, patients, and physicians. Manufacturers can use their resources in more efficient way because analytical study can require less time because instead of patient samples, sometimes can be used surrogate sample. There is no need to repeat some analytical studies because of inappropriate surrogate sample preparation. It is important to note that when the surrogate samples are prepared in such a way that reflects the patient sample in the best possible way, the estimate of the analytical performance provides information about the assay performance with the patient sample. Otherwise, the performance estimates can be biased, usually overstated. All this leads to a faster and more consistent reviews. It is beneficial for patients because of faster access to good laboratory tests. It is beneficial for physicians because information in labeling about assay performance based on analytical studies which use frequently surrogate samples reflects the assay performance with the patient sample in the best possible way. Details of the surrogate sample framework will be presented by Dr. April Vukas. Next slide.
Thank you, Marina.
We're done.
In regards to the use of surrogate samples, there were a number of items that prompted the team to focus in this area. Next slide. As noted by Marina, absent guidelines, the use of surrogate samples may result in regulatory uncertainty, repeat studies, and inefficient use of biological test materials. When actual patient samples are in short supply, the use of surrogate materials or samples provide a source to develop innovative and improved diagnostics, increasing patient access. The Surrogate Sample Work Group established a foundational framework to promote efficiency and improved dialogue when surrogate samples are used to support IVD validations. Next slide. This slide acknowledges the individuals who were a part of the work group that developed and published the framework. As you can see, we have individuals from industry, government, as well as expert advisors, and I personally want to acknowledge all the contributions of the group to this final product. The draft framework was open for public comment, and we did receive very thoughtful comments, and these comments were used to help inform the final framework. Next slide, please. Early on, our team conducted a survey of stakeholders and research to help identify the variety of reasons for using surrogate samples. Surrogate samples provide an option when naturally occurring specimens of biological origin are rare or difficult to acquire. This, for example, would include supplementing patient specimens across the entire range of an assay. For example, the need to have more samples at the high or low end, or the medical decision point may prove challenging when there is a low prevalence of these specimens in the general population. Surrogate samples can help address these challenges. Ethical considerations is another reason individuals may turn to the use of surrogate samples. For example, invasive sampling methods or drawing large volumes of clinical specimens from sick patients are another reason surrogate samples are used. Additionally, surrogate samples may help simplify sample acquisition and characterization activities to meet scientific demands of certain studies. For example, when large volumes of sample are required. Next slide, please. Before discussing the details of the framework, I wanted to spend a few minutes describing what the framework is and what the framework is not. First, one of the key objectives was to establish a common language to facilitate and guide discussion in this area. There are a number of terms that have been used: surrogate, contrived, altered, and pooled, and aligning on definitions and terminology is a key element of this framework. The team also looked to strike a balance between general information to guide decision processes related to the use of surrogate samples, as well as recognizing that there are some exceptions or alternatives that would apply to certain study types. And although the framework contains information related to a particular study type, for example, linearity or precision study, the information here is related to the use of surrogate samples. It's not intended to provide all the elements of study design that one should consider. And the reason for this is this information is available in existing standards and guidelines. Lastly, the framework centers on a hierarchical approach to the selection of the appropriate type of surrogates for use in a particular study type. Next slide, please. The framework contains the following sections or elements. It creates a uniform definition of surrogate sample, which is also supplemented with a diverse array of examples. These examples can be found throughout the framework, as well as additional information in the appendix to the framework. The framework outlines general principles and considerations when designing and selecting surrogate samples, so a broad view of items related to studies across the different study types. It presents a hierarchy to aid in the selection and design of surrogate samples. It also establishes study-specific principles and is accompanied by a hierarchy for each study type. Next slide, please. When our team first started, we looked for an established definition of surrogate or contrived sample. We looked in standards and FDA guidance, but we were unable to locate one. Because different terms were being used, contrived, altered, spiked, this actually hindered early discussions within the work group, and we recognized a need for a common language. Next, we started to look for related terminology that would be defined in standards, such as the term surrogate material, to create the following definition of surrogate sample, which is material or combination of materials that is used as a substitute for body fluid or tissue taken for examination from a single human subject to study the characteristic of interest. We supplemented with the definition with examples to provide further clarity. Equally important, though, is what is not included in the definition, which is preparing an analyte for measurement with collection or storage media or pre-analytical steps does not meet the definition of surrogate sample. So this would still fall under the definition of patient sample. So this definition is not intended to include the pre-analytical processing steps that are part of an assay that does not convert a patient specimen into a surrogate sample. Next slide. To provide additional clarity within the definition, surrogate samples are further described as falling into one of three categories: supplemented, pooled, or simulated. Supplemented means a matrix that's been spiked with a target analyte, while pooled refers to the combination or combining of individual specimens, and these may or may not be spiked with a target analyte. And then simulated refers to altered materials of biological origin, such as biological material that's been heat treated, for example. And it also includes artificial materials created to have the properties similar to or representative of a body fluid or tissue. Next slide, please. This framework establishes a table of surrogate sample combinations based on matrix, which is across the top of the table, and analyte on the left side of the table. In the broadest sense, a matrix may be biological or artificial. And artificial meaning a matrix that has been created with the intention to stimulate the specimen biological matrix. Matrix is further categorized as unaltered or altered. And then within those categories, you may have individual samples or pools created by combining one or more, or two or more individual patient samples. In terms of analyte, the table focuses on the analyte that may be spiked or unspiked, and then further breaks it down into biological or artificial. The table, as you can see, has alphanumerical identifiers assigned, and this is to make it easier to refer to the different types of sample combinations throughout the framework. It has no additional meaning other than that. It's a means to create a uniform way of referencing these samples throughout the framework document. Next slide, please. The framework sets forth general principles to assess and identify the factors most pertinent to the study in which the use of surrogate samples is being considered. So initially, it is recommended that one first clarify the objective for using a surrogate sample. Is it to supplement naturally occurring specimens, or is it intended as a replacement for the naturally occurring specimens? The next general principle relates to identifying critical factors related to the performance study itself. What is important in terms of that particular study, and how does one best achieve that with a surrogate sample in terms of the ability to mimic biological variability found in individual specimens, to meet scientific demands of a particular study, and to take these factors in consideration when selecting and designing a surrogate sample. Next one would consider factors based on whether the test is qualitative or quantitative. Next slide, please. Another general principle is to consider the effect of sample processing, such as an extraction method or other pre-analytical treatment. Consider which property characteristics of the naturally occurring specimen are most relevant, and then recognize that the surrogate sample may be different for different types of studies. Next slide, please. This slide presents the surrogate sample hierarchy. In the presentation, the surrogate sample hierarchy is presented on 2 slides. So this is the first of the 2 slides. This part of the hierarchy focuses on where to start, with the green box here representing an individual patient sample. So in a perfect world, one would have an individual patient sample as the test material for the particular IVD. In this case, patient sample includes prospectively collected and tested samples, prospectively collected but retrospectively tested samples, as well as archived samples. Additionally, as noted before, this also includes specimens that have been collected or stored in media or that have had pre-analytical steps to prepare the analyte for measurement. So with this as our starting point, when these types of samples are not available and one is looking to use a surrogate sample, the first decision point is either to use a pooled surrogate sample or an individual sample spiked with an analyte. And depending on the study type, the type of surrogate will vary, and this is contained in further detail within the framework. In addition, as we look at this hierarchy, there's a general movement from the left side of the hierarchy to the right in terms of order of preference. So although there may be different factors to consider within this particular study type, as a general rule, if we're looking at the hierarchy, we're moving from the left to the right. Next slide, please. This is the second part of the hierarchy, and it's a flow chart that identifies preferred approaches related to dilutions in order to minimize the effect of dilutions on the preparation of the surrogate sample. And it also includes information related to spiking of materials in order to preserve matrix integrity. Next slide, please. Next, the framework moves from discussion of general principles to study specific principles. There are 9 different performance study types addressed within the framework, and you can see these here on the slide. For each of these study types, there is a narrative that describes principles to aid in the selection, and then there is also a study-specific hierarchy that is included. Next slide, please. This is the table of the study-specific hierarchy. For each study type, there is a column. So the top row describes the different study types. And then there is an order of preference presented underneath each study type. So each column is showing an order of preference in relationship to the different types of surrogate sample combinations. So rather than using the words, we use the alphanumeric identifiers here. And so these do correspond back to the combinations table that was presented earlier. And the shading represents different types of guidance related to the particular study type. So generally, as we've noted in previous slides, patient sample is the place to start. And this is represented by the darker green shading in the top row. Next, the medium green shade is used to indicate those study types where there's a downward linear progression of selection of the surrogate sample type. So one would want to start closer to the top of the column in terms of selection of the surrogate, that these combinations have been determined to be the most appropriate for the particular study type. In other areas, which are reflected here in a lighter green shading, the decisions are fact-specific and may depend on different factors, such as the assay type or the sample type. So, you know, is it a chemistry assay or is it a molecular assay? There are different factors that need to be considered, and it's not necessarily the same sample type for each of these assay types. The other shaded color reflected in this table is the peach shading, and this really recommends areas where the work group is indicating proceed with caution and really explore these areas only after exhausting the other options. So as one moves through the selection process and determines, you know, scientifically that there's a reason not to use a particular sample type, when you venture into these areas we've indicated with peach, there's less backing in terms of experience in terms of the use of these types of samples for the particular study type. And when looking at this, really consider your scientific rationale for moving in this direction. A few of the columns end with an asterisk, and these are areas where, based on the experience of the work group and the information collected, that the other sample types may not be appropriate for the study type, and wanted to ensure that we had a way of indicating that as part of the framework. Next slide, please. So in terms of accessing the framework, it is available on the MDIC website. There is a link presented here to aid you in accessing that framework. As it relates to the particular work activities of the work group, the team is now moving on to the development of more detailed training material to accompany the use of the framework. And as part of that, looking at case studies to include within that training material to further illustrate the concepts and application of the framework to actual situations. I'd like to now move us into the questions portion of the webinar, and you can type in your questions for the panel in the chat box in the lower right-hand corner of your screen. And I will now turn the webinar back over to Carolyn.
Hello, it's Marina Kondratovic. Let me read first question. Did you consider the effect, oh, I lost. Okay, just a moment. Did you consider the effect of cross-blood effect, if any, when pulling blood from multiple donors, and its potential impact of test of interest? Wouldn't it be the supplemented sample better than pooled sample for most of the tests? It exactly depends on the what kind of analyte, what is the test, and you see that in both your approaches can be valid for different situations for different type of the test technology. And also, of course, for the what kind of analytical study you are evaluating. For example, for comparison study, probably it's better to have individual sample and to spike. But for example, if we consider linearity study, in linearity study you need to have intermediate sample, and you still need to pull high sample with some low sample. So you still use some pooling of sample in order to prepare intermediate. So maybe even high sample can be also pool sample. So you see, it depends on what kind of study and what kind of test you have.
Thank you, Marina. Can you hear me, Marina?
Yes.
Okay. We have some additional questions here, and I certainly have a gremlin in my system right now, but another question is, how do the definitions of specimen and sample differentiate? How are they different?
Maybe April, yes?
Sure, I can take this question. So in terms of the framework, the term specimen is being used to refer to a patient specimen, and within that category we are talking about prospective, retrospective, and archived within the definition of patient specimen. And then the term sample is a broader use term and could encompass patient specimen as well as the surrogate sample as well.
Another question. Is spiking whole blood before converting to plasma or serum considered an acceptable form of surrogate, or it is preferred for spiking to occur directly into plasma or serum? So in order to answer this question, let us use our basic principles that we would like that surrogate sample mimic patient sample as much as possible. So if you have some kind of steps in order to prepare your sample and then put in your testing system in order to obtain results, then of course we would like to mimic patient as much as possible. So when you really spiking in the whole blood before converting to plasma and serum, you like creating some artificial patient. And then all your sample is going through all this pre-analytical step in order to prepare, for example, plasma. It's definitely for a lot of, I cannot tell that for all, but for a lot of tests, probably it's better mimic patient sample than to prepare plasma and then to spike. But maybe there are some unique situation. I don't, of course, again, I cannot tell for 100% of all tests, so maybe there are some situations when it will be okay to spike directly into plasma or serum, but definitely using our principle, it's better to spike and then to prepare serum or plasma. Maybe some difficulties when you're doing this step. Please pay attention that every time we tell them that it's possible to do, it's the best available method. So if it's difficult to do, there are some other issue, maybe with some stability or something, then maybe you can also spike directly into plasma. But my answer, probably first approach what is described is better.
Thank you, Marina. What are the key challenges IVD test developers face when they're using surrogate samples?
So this is April. In terms of some of the key challenges, one is just the selection, and the framework is intended to help put together more of a thought process to aid in that selection. In terms of preparation, that is another challenge, and the framework does address preparation issues related to matrix and dilutions in order to help provide some more consistency in terms of that aspect. The other piece is really to focus on the rationale for choosing a particular type of surrogate sample, and focusing on going through that thought process and that scientific analysis in terms of selecting the appropriate or the best surrogate for that particular assay in that particular study in order to avoid the challenge of having to conduct repeat studies.
Okay. What do you think are the most important elements of the framework?
So this is Giovanna. I think April described really nicely what are the contributions that we thought we were adding with this framework. One of them being the definitions, and I think April went through specimen versus sample that somebody had a question. Because there are so many different definitions and so many different terms that people are using to express the same thing, it gets confusing. So we're trying to streamline some of that. And the other part is to show the hierarchy. First of all, that surrogate samples can be used, and maybe it can be preferentially used for some of the studies. And the second, to have the hierarchy of which studies should be using surrogate samples and what surrogate samples, and then which studies should probably use clinical samples and not surrogate samples. So I'll throw it over to Marina and April too.
I don't think I have something to add because I completely agree. I think that a lot of very great points, and I think that this document will help to find manufacturers to find some kind of rationale why they selected this particular type of the surrogate sample, and help them to streamline their thought process, and maybe they will realize that this is not the best surrogate samples preparation, so they can do better. They can have better estimation of the assay performance with for patient samples. So the closer to the patient samples, the better.
Okay.
Mm-hmm.
Does this approach work for all types of tests, automated as well as manual, immunology and microbiology, for example?
So I'll give it a short answer because that's a very general question, and we also have another question that we can see in the chat. So definitely can work for all different tests, but we have been very careful not to be too prescriptive because there might be cases where you can be best served to go to specific. If you're coming to the FDA, if you're not in Europe, if you're in the U.S., to come and ask the specific division better. This is the approach that we are proposing to take, and ask them whether that would be okay with them, for instance, for microbiology or immunology.
Great. Now, Giovanna, you mentioned that you can see the chat box. I want to apologize.
Yes.
My system has froze up on this end, so hopefully things are still going okay on the screen. Marina, or if you can see the questions in the chat box, would you be so kind as to answer them, because I'm not able to scroll to them.
Also, will it contain examples of accepted rationales? But I think it's already, you answered, yes, what kind of different data and studies? So, Giovanna is answering
let's go to the first. So the first question was, will the guideline framework contain examples for surrogate samples for different assays and studies? So the guideline or the framework as it's written, as you can see it right now, is done for now. Now, in the training, or maybe somebody else can take that framework and further develop it. Maybe they can have examples. But the framework, and April can correct me if I'm wrong, is as is right now. It's been published as is. And then Marina.
Yes. Also, will it contain examples of accepted rationales for the use of surrogate samples? Will the framework be integrated in the accepted standards at the FDA? With regard to accepted standards at FDA, yes, we will make reference to in our FDA guidances. We will have training for our reviewers how to use these principles described in the surrogate sample framework. About whether we're planning to have examples, more examples of accepted rationales for the use of surrogate samples, I think that maybe Carolyn will help me if we, my understanding that we're planning to have maybe some documents from Clinical Laboratory Standards Institute, and maybe if it will be, this project will be accepted, maybe it will be some appendixes with more examples for some particular situation, for some particular technology, some particular type of the test. But again, this will be only example. It's not easy to write a lot of details for some particular example because it can mislead people in other situations. They can think that it's really very similar, but in reality maybe there are important differences that this example may be not acceptable. But we think that better to use general principle from the beginning to your particular situation and to put on the table your rationale why you think this type of a surrogate sample is acceptable for this study and for your technology. Not try to copy some example for different technology and for different study. This is my advice.
Yes, and just to add to that, the framework is taking this general approach in terms of application to surrogate samples, and then getting specific when it comes to study types, as in many cases there are some overlapping situations regardless of the test type when you're considering a particular study type. The framework also does include useful information as it may relate to particular situations. You know, for example, in terms of mimicking, patient specimen, there's some discussion about, the viscosity of the matrix. So this is contained primarily as examples within the general principles, but for certain situations, these examples were intended to help supplement real-life application of the points to consider.
Okay, are there any more questions?
Not so far. Not so far, at least in the chat box.
Okay. Then I think what we'll do is we'll wrap up. And again, I'd like to thank the Surrogate Sample Working Group as well as the panelists.
There are 2 more questions now coming in.
Okay. All right. Have at them. I can't see them because my screen froze up.
Yes. Have you addressed this issue in the pediatric population, especially neonates? In this framework, this particular issue was not addressed directly, but I hope that basic principles still can be used, and this is interesting topic for future work. How, in which situation we can use surrogate sample for pediatric population. Like for some studies, maybe it's impossible. Like, for example, I think that if you speak about reference interval, I did not see any possibility how you can do this. but some other studies probably yes. Another question, what is expected for the level of proof that more preferred surrogate sample types are not appropriate for a given application when submitting data based on less preferred types? I think that, this framework, it exactly will be very helpful in this situation because you have a hierarchy of the samples. Then you see that what kind of samples theoretically can be considered better, and for this particular type of the sample, you provided your rationale, or did you try to do this, or why you think it's even completely impossible to do this, or maybe it's really for your particular situation, what you proposing, it can be better because how April described, there are some cells which are the same color. So inside of these cells is the same color, like for example, light green. It can be a different hierarchy, yes, like for some situations it can be maybe matrix more important, for some maybe analyte. So this document will help you. So please look at the this document. It will help you to describe type of surrogate sample which you not use. But of course, details why it's impossible to prepare. This depends on your situation. You know better your device, and analyte, patient sample, what kind of characteristics they have, and what characteristic important for your study. So this depends on your situation, but I think a framework will help you to answer this question.
And then we have the last question, and that's somebody who seems to be a volunteer to get involved with this committee. So maybe Carolyn or April can give some idea of that. I don't know whether this specific committee surrogate samples or in general.
So Carolyn maybe did not see this text.
Okay.
How can one get involved with this committee?
If someone is interested in becoming involved in one of MDIC's activities, please feel free to send me an email, and my email address is chiller@MDIC, that's CHILLER @MDIC.org. I will happily share with you how you can get involved in our initiative. And I want to thank everybody for your patience, and our working group as well as our panelists, Annie Saha, Marina Kondratovic, Giovanna Tezak, and April Viokas. You can download the framework on our website, and a recording of this webinar will be available for viewing in the near future. Thank you very much, and I wish you all a wonderful day. Bye.
Thank you. Bye-bye.